Okay, welcome to today's BSWG KOL series. And I'm really delighted to introduce our returning guest speaker, Dr. Yuan, who needs no introduction. He's from MD Anderson Cancer Center. Dr. Yuan is a Bayesian Ashley Murray Distinguished Professor and Deputy Chair in the Department of Biostatistics at University of Texas MD Anderson Cancer Center. Dr. Yuan is an internationally renowned researcher in innovative Bayesian adaptive designs, including early phase trials, seamless trials, biomarker-guided trials, and basket and platform trials. The designs and the software developed by Dr. Yuan's lab, trialdesign.org, have been widely used in medical research institutes and pharmaceutical companies. I know I use that website all the time. The Boeing design developed by Dr. Yuan's team is a groundbreaking oncology dose-finding design that has been recognized by the FDA as a fit-for-purpose drug development tool. Dr. Yuan was elected as the American Statistical Association Fellow and is the leading author of 2 books, Bayesian Designs for Phase 1 to 2 Clinical Trials, and model-assisted Bayesian designs for dose finding and optimization, both published by Chapman and Hall CRC. So without further ado, Dr. Yuan, the floor is yours.
All right, thank you. Good morning. First, thank BSWG for the invitation and for this opportunity to share some of our recent work on this very hot topic, dose optimization. Okay, so this is a joint work with my PhD students, Pen Yang, who is at the Rice University, and my collaborator, Daniel Lee at BMS, Ray Tao, my colleague at MD Anderson, and Bo Huang at Pfizer. Okay, as we know, FDA in 2020, FDA launched a Project Optimus to reform the dose optimization and dose selection paradigm in oncology drug development. So this is an important initiative and affect many aspects of drug development, especially for oncology drug development. So the reason for this initiative is the paradigm shifting from chemotherapy to target and immunotherapy. So here contrast the characteristic of those 2 different type of therapies. So on the left, that's the conventional chemotherapy. And you can see the x-axis is the dose and the y is the response, including efficacy and toxicity. So for chemo, most of the time when you have higher dose, you will see a substantial increase in toxicity and efficacy. And in this case, maximum tolerated dose basically is the dose that acceptable and deliver the highest efficacy. So in this case, MTD essentially is the optimum biological dose. And a lot of design has been proposed that's conventional so-called dose escalation trial. We escalate dose until we reach the MTD. And most of the time, we're going to use that dose as a recommended phase 2 dose in a phase 2 and 3 trial. And on the right side, it's kind of more recent, drug development kind of field, and we have targeted therapy. And it's quite different from conventional chemotherapy. And you can see targeted therapy often has a wide therapeutic index. Toxicity increases with the dose, but efficacy may plateau when the dose reaches a certain level. So as shown in this graph, and you can see the efficacy actually reaches the plateau before reaching the MTD. And in this case, OBD, optimum biological dose, certainly it's more favorable and a better dose for a treating patient. So in this case, safety alone, it's not sufficient to inform the optimum recommended phase 2 dose. So this is the reason, I think behind the Project Optimus, why we should shift from MTD-based dose finding identification to the optimal biological dose identification. So there's a different design strategy to find optimal biological dose. And this also depends on which phase you are. There's a phase 1-2 design, there's a phase 2-3 design. And in the context of early phase 1-2, and in general, design can be classified into two different strategies. So one, I call it efficacy integrated dose finding strategy. So in this strategy, what we do is we have toxicity efficacy endpoint, and we have some criterion to measure the desirability of each dose in terms of benefit risk trade-off. And based on the data, we continuously update our estimate of desirability of each dose to guide dose escalation, de-escalation. And the basic idea is very similar to all the dose funding, especially model-based dose funding design, like the CRM, right? So we treat a cohort patient, we get new data, we update our estimate, make a decision of dose transition, escalate, de-escalate, or stay. The only difference, different from the CRM, is in this case because we consider both benefit and the risk. So we won't only focus on DLT. We will consider both toxicity and efficacy and come up with a metric to measure the desirability. And we estimate the desirability to make the decision of dose transition. So in the bottom, I showed some example of this efficacy integrated dose finding strategy. And then other strategy is, I call it two-stage dose finding strategy. So in this approach, we kind of conceptually have two stages. The first stage is dose escalation. And this is almost the same as the standard dose escalation design. So we focus on toxicity. We, through the dose escalation, identify a set of safe doses set. And then in the second stage, we're going to further study the efficacy, toxicity, tolerability of more than one dose. And typically, we will move MTD and one dose lower than the MTD and randomize patient in stage 2, so we can get more understanding of those two doses. Eventually, we can pick the optimal dose. And you can see in this strategy, so in stage 1, it's pretty much the standard dose escalation. So we can use all the design we have, right? Like the CRM, Boeing, or even you use 3+3. So that's the stage 1. So in this approach, the real challenge is the second stage, right? First, how we determine sample size, how we randomize, how we make a decision. So that's the focus of this talk. So we're trying to come up with a design and in a principled way to address the second stage. So let's first see how FDA guidance talk about this issue. So this is a new guidance released January this year. And the guidance have a specific section talk about trial designs to compare multiple dose. And the goal is to compare in the multiple dose in a trial or to decrease uncertainty with identification of optimal dose. And specifically, the guidance talk about recommend use a randomized parallel dose response trial, which is essentially the same as I illustrated in the previous slides. We randomize a patient among multiple doses. And further, the FDA guidance give 3 points as showed here. And you will see the first point is basically provided the kind of a rationale for a randomization, because it's going to ensure similarity among patient. So that's going to facilitate the comparison and interpretation. And from a design perspective, the second point is it's more relevant. Okay, so if you read this, this is a very thoughtful kind of recommendation. The first is it's kind of give a upper limit, upper bound of sample size. So you read the second paragraph, a second sentence, it says, the trial does not need to be powered to demonstrate statistical superiority of a dosage or statistical non-inferiority among the doses. So the reason is if you do calculation, you will notice that the sample size is going to be huge to demonstrate superiority or non-inferiority. So basically, this give us the upper limit, okay? And to do the trial, we do need more specific guidance, right? How to choose sample size. And the guidance gives say the sample size should be allowed for sufficient assessment, activity, safety, and tolerability. But what you mean sufficient? Is there any statistical criterion we can use to gauge that sufficiency? So that's the problem we want to address in this design. And the third point mentioned by the guidance is about adaptive design to stop enrollment, use monitoring, toxicity or futility monitoring. So in the merit design we're gonna present today, we also want to address this issue. So when we work on this, we start to realize, so this is a really complicated problem. So the reason is dose optimization and the selection OBD, it involves so many different data and factors. So if you look here, it says the activity, safety, and the tolerability. And for each of those things, there's many data, right? For activity, there's all kinds of activity. There's a clinical activity, there's biological activity. So it's involved a lot of data and the totality of the evidence. Same about the safety and the tolerability. So this makes it almost impossible to fully quantify this objective. And without that, we really cannot make much progress come up with a statistical criteria. So what we do is we trying to separate the aspect we can quantify and use the statistical criterion and separate from those which it's better or more reasonable based on empirical decision. So that's why we come up with this two-stage decision-making paradigm for dose optimization. So basically, there are two steps. The first step is determination of the OVD admissible set. What that means. So in this step, we try to identify a dose that satisfies certain safety and efficacy requirement. And we call this admissible to be a OVD. And here we use A denote that admissible dose set. And this we're going to based on some pre-specified toxicity and efficacy endpoint. And we will talk a little bit about more how to select the toxicity and efficacy endpoint in following slides. Okay, after this step, we have an admissible set, which can be selected as an OVD. And then in the second step, we're going to identify the OVD. And this identified OBD, so FDA guidance say it's relevant non-clinical and clinical data, as well as the dose exposure response relationship of safety and efficacy should be evaluated to select a dose for clinical trials. And you can see this involves a lot of consideration, totality of data, statistical and non-statistical consideration. So we feel it's much reasonable to leave this step as kind of empirical based on team discussion, statistician, clinician, and also pharmacology scientist to make this decision. It's very hard to come up with, a statistical criterion to pinpoint this specific OBD. So that's why we take this approach. And because of that reason, we're going to focus on the first stage, because this is a stage we can really quantify and ensure the property and accuracy of this decision-making process. So our goal here is to formalize the design of this step to ensure that the trial satisfies certain statistical property, specifically type 1 error and the power. Okay, so here is the setup, and then we can introduce how we do it. And we consider a multiple dose randomization trial. And we have J times N patient are equally randomized into J dose. And in most of the applications, J typically is 2 dose or 3 dose. And the highest dose is often the selected MTD from the dose escalation study, or maximum administered dose if the MTD is not reached. And here we use YT and YE denote a binary toxicity and efficacy endpoint. So those two endpoints should be selected based on the trial characteristic and objective. So for example, for the toxicity endpoint, it can be a conventional dose-limiting toxicity, and it can be also, if for some target agent, we don't expect any DLT, And in this case, we may consider the low-grade toxicity. For example, we can use a total toxicity burden. And because in here we focus on binary endpoint, you can think we can dichotomize into a high burden and a low burden. And it can also be a dose tolerability. So for example, it's the rate, how many, the percentage patient discontinuation, reduction, or interruption. And this is an important factor in many target agents. So typically for target agent, it will be administered multiple cycle, and tolerability actually is more of a concern than a DLT. But in this design, we can take that into account. Okay, so it's just toxicity endpoint choose based on the trial and the drug characteristic. And for the efficacy endpoint, it can be objective response, and it can be also some efficacy surrogate endpoint. For example, if we have a good PD endpoint, want to take into account, or some other surrogate like target receptor occupancy, those can also be used as efficacy endpoint. Again, here what we do is a randomized trial, right? So you know, the assessment period, it's not that critical. For those funding trial, a lot of time we require the endpoint needed to be assessed quickly, so we can make an adaptive decision for each cohort. But in this case, we are in the context going to randomize patient in multiple arms. At the end of the trial, we make a decision. So in this case, we have more flexibility to account for clinical endpoint of interest and importance. So here we use pi TJ and pi EJ denote the toxicity and efficacy probability for dose DJ. And in here, we assume pi TJ and pi EJ are non-decreasing with respect to the dose. But this assumption, it's not required by our methodology, but in most cases, this is true. That's why we focus these cases. Okay, so and then we have two kind of hypothesis rate, right? So here the phi T0 and the phi T1 denote non and alternative toxicity rate. So phi T0 means toxicity rate is high, that deems unacceptable. And the phi T1 is alternative toxicity rate that is low and deemed acceptable. And similarly, we can define phi E0 and phi E1, which is non and alternative efficacy rate that deems unacceptable and acceptable respectively. Okay, so with given the phi, acceptable, unacceptable rate, and we can define a dose is admissible or not. So this is immediately leads to a 2x2 table, right? So toxicity, the red one, phi T0 indicate unacceptable. Green one means acceptable. So we use the same color for the efficacy. Red is unacceptable. Green is acceptable. So in this case, a dose it's acceptable only when both are turned green. So as shown in here. Now, so we formed two hypotheses. Okay, that's how we can rigorous define the type I error and the power to ensure operating characteristic of the design. So in here, the non-hypothesis is none of the dose is the OBD admissible. Okay, so in here I didn't put the admissible. Basically, it's not, if it's none of the dose, it's OBD, so obviously it's not admissible. Right, so same thing. So alternative is at least one dose, it's OBD admissible. And we will first consider type I error and then power in following slides. Okay, so we first think type I error. Of course, this is under the non-hypothesis, right? So in this case, the challenge is different from a simple, for example, we have two-arm comparison or we have single-arm trial. In here, a challenge is each of them, each arm is a dose, right? So in this case, our goal is to select the OBD admissible dose. That's why in this case, the real challenge is non-hypothesis. It consists of multiple hypotheses. So for example, when we have two doses, D1, D2, as shown here, actually we have 6 hypotheses corresponding to this H9. And for example, if you look at the first one, let me use. So if you look at the this first one hypothesis, So you will notice in this case, both those two are unacceptable in terms of toxicity, although the efficacy is acceptable because they are green. So in this case, both doses are not acceptable. So this corresponding to the non-hypothesis. None of the dose is the OBD admissible. But of course, there are different combination can lead to H-non, right? So for example, in the second case, and the drug is not safe for both doses, but high dose is efficacious. But again, because high dose level 2 is toxic, it's still not acceptable or admissible in terms of OBD. And if you enumerate all those possibilities, you will get a multiple hypothesis. And specifically, so if we have capital J doses, so this is the number of possible non-hypotheses corresponding to H-non. And what we want to do is we want to control the type error for all possible non-hypotheses. So to towards that goal, and we define a global type error to encompass all the H-non. And so basically, we can define the type error as, so the first equation is a typical definition of standard definition of type error. Given none, what's the chance we reject none? But I think the difference right now is we have considered all possible non-configuration. We take the maximum one as the global type error. And we want to control, in the worst case, the type error is controlled. So that's the basic idea here. Okay, and once we have type error, and then we can see how we define the power. And it's not hard to imagine, similarly, H1 going to encompass a collection of hypotheses, right? So for example, we may have two doses, D1 and D2, and those are three possibilities which corresponding to the alternative H1. And for example, in this first hypothesis, and those two, both are efficacious, both are green. But in terms of toxicity, only the lower dose is acceptable. High dose is too toxic. So in this case, D1 is OBD admissible, right? And you can see the second scenario is both are admissible, both are green. And the third case is only in the higher dose admissible. And if we write down, go through the notation exercise, so basically you can enumerate, write down in this way, and essentially where it is the acceptable and unacceptable toxicity and efficacy. If you go through the exercise, you will come up with this conclusion. It's going to have a summation of J of hypotheses. And there's another challenge associated with power, okay? Because in this case, when we reject none, it doesn't mean we did a good job, okay, for the purpose of dose optimization. So again, so this is a different from a typical, hypothesis testing, single arm and the two arm trial. So for example, I give a very simple example. Let's say the truth is dose level 1, the low dose is safe but futile, and the high dose is safe and also efficacious. We can reject none and conclude dose level 1 is admissible, right? So that's a reject none. Reject none is correct because there is one dose, so D1 is good or D2 is good. But the problem is if you reject none, select D1 as OBD admissible, that's incorrect decision. So recall our two-stage OBD selection framework. First stage is to identify the OBD admissible set, basically the set of dose that satisfy pre-specified toxicity efficacy criteria. And then in the second stage, we're going to based on totality of data and also some non-clinical consideration to select a single OBD that's recommended phase 2 dose, going to use in a phase 3 trial or phase 2 trial. Okay, so that means the second stage decision will rely on the first stage. And if in the first stage we select the D1 as OBD admissible, and then in the second stage when we expect the totality of data, we may say D1 is not qualified. And in this case, we made a wrong decision, right? It's going to lead to failure. So that's why in additional to consider the probability to reject none, we also have to account for the quality of the admissible dose selection. So that's another challenge. That's also a difference from the standard definition of power. So towards that goal, in here, we propose two generalized power. One is called generalized power 1 and generalized power 2. Both are taking into account the OBD admissible set selection criterion. Okay, so the first one is we reject none. At the same time, we also want all doses in A are truly safe and efficacious. So recall the A indicate the set of dose we selected as OBD admissible. Okay, so all those in the admissible set are truly safe and efficacious under the alternative hypothesis. That means there is some dose which is truly OBD admissible. And the second definition is a less strict. So it's required at least one dose in the A is truly safe and efficacious. And for each definition, we can define the global power 1 and the power 2. Again, the reason we define global is because we have multiple, under the alternative H1, we have multiple hypotheses corresponding to the H1. So that's why in here, we take a minimum one, most conservative one. So if that power is controlled, every scenario, it's controlled. And here is a little bit more comments in terms of those two generalized power. And first is both generalized power are stricter than the standard power. So that's very obviously, because in addition to reject none, it's have additional requirement. For example, all select dose is truly admissible, or at least one of them is truly admissible. And as I just mentioned, so this additional requirement is to ensure the quality of subsequent final OBD selection, right? Because if you only you select a wrong admissible set, and then from that set you're going to fail, you cannot select the true OBD, which is stage 2. And also generalized power 1 is stricter than generalized power 2. And the choice of which power in your trial is going to depend on the trial characteristic and user's tolerability of false positive. And in general, we feel under this two-stage decision-making paradigm, a false positive it's of less concern than standard hypothesis testing, because the false positive made in step 1 could be identified and corrected in step 2 based on more data. Again, in stage 2, we were based on a lot of data, right? You know, even for toxicity, efficacy, it's just one endpoint. It's based on multiple multiple dimensional safety data and also efficacy data and also tolerability. In addition, we also have PK, PD, and also biological activity data to make the second and the final decision, which dose is the OBD. So in other words, in the second stage, actually we have more data, more information, basically it's higher power eventually to identify the OBD. So in the stage 1, if we make some false positive, most likely we are captured in the second stage. So that's for that reason, we think if reduce the sample size, it's top priority, and generalized power 2 may be a good option. Okay, one interesting about this setting is we said for power there is multiple configuration or hypothesis corresponding to the alternative, right? And one interesting statistical property is here we can identify least favorable set. So basically, if you look at here, the least favorable set, look at the definition, H1J. So essentially, it's only one dose. It's safe and efficacious. So you have multiple doses, right? So there's maybe 2 doses are safe and efficacious, or 3 doses are safe and efficacious. And this least favorable set is only have 1. And it can be shown, so this case is going to have the lowest power. And so the reason we have this property is it will make definition and eventually optimization easier. Because in this case, we don't have to enumerate both location. If you look at the original one, you have UV, both have to enumerate. But in this case, because we only pinpoint, there is only one OBD. So in this case, there is only J possibility, and which is a decrease dimension. You think about the case, we have 2 doses, okay? And when you have 2 doses, we showed at least there's 3 possible alternative, right? And 1 dose, dose 1 is admissible, dose 2 is admissible, both doses are admissible. So that means you have to enumerate all 3 possibility, control the one have lowest power. But this least favorable set says we only have to consider 2 configuration. Dose 1 is OBD or dose 2 is OBD, because those 2 we have lowest power. It's going to happen in one of them. But for 2 doses, it doesn't, it's only reduced from 3 configuration to 2. But when you have 3, that will decrease furthermore. Okay, so we are going to present with the definition of global type 1 error and global type 2 error 1 and 2. And we are in the position to introduce the design we call the merit design. Okay, multiple dose randomized phase 2 trial design. And for simplicity of time, I just say type 1 error, type 2 error, type 1 error and power. But basically it means the global type 1 error and global type 2 error, either 1 or 2 defined previously. Okay. So in this design, it's pretty straightforward. The first step is we specify target global type 1 error and the power, alpha star and the beta star. So this is no different from any study design. We have a type 1 error power, a target value. And the second step is we're going to randomize J times N patient equally into J doses. Okay. And in any dose arm DJ, if NEJ greater or equal than ME and NTJ less or equal than a certain threshold, we're going to reject none and claim that this dose is OBD admissible. So in here, the NEJ is number of patients who experienced efficacy. This number of patients experience toxicity. So basically, this criterion says the number of toxicity is greater than certain threshold and toxicity less or equal than certain threshold. We're going to reject none and claim this dose is OBD admissible. So you can see this is an extremely simple design. And that's one very important factor. I like this design because it's simple. Okay, so when you do it, this is really not any difficult things to do and implement. Okay, so in this design, the only parameter we have to determine is the N, right? Because how many doses we already designed after that dose escalation, we designed in this stage, we're going to, for example, choose 2 or 3 doses. So we have to determine N, how many patients we randomize to each dose arm. And the second is what's the efficacy threshold, what's toxicity threshold, MT and ME. And how we do it. So in here, it's pretty simple. We can do determine those 3 design parameters through a numerical search, such that the design controls a global type 1 error and the global power at a nominal value. And we, at the same time, we reach that threshold with the minimum sample size. And here, this is not, we just showed the formula, essentially we can calculate. So it's not a key point here. Okay, so this shows the sample size and decision boundary for one case when we non-toxicity alternative toxicity rate is 40% and 20%. This says 40% rate. And similarly, this is shows acceptable and unacceptable efficacy rate. 20% response rate is not acceptable. 40% is acceptable. Okay. And here we listed 2 dose case and 3 dose. So we have 2 dose randomization, 3 dose randomization. And this column beta star shows our target power, and 60%, 70%, 80%. And on those columns shows the for different type 1 error, like 10%, 20%, 30%. So for example, a priori, we want to do a trial which has 2 doses. Okay, we want to control type 1 error 20%. We want to control power 70%. So in this case, Under the Marriott design, it says we need to randomize 25 patients to each arm, okay, 25 per arm, okay, totally 50. And the threshold for toxicity is 7, and efficacy is for 8. That means when you randomize, collect the data, you count the number of efficacy, number of toxicity. If in any dose arm, if number of toxicity less or equal than 7, at the same time, number of efficacy greater or equal than 8, we're going to claim that dose is OBD admissible. So that's the design. And this shows under different scenario. And you can see in most cases, the sample size, it's not that big. It's reasonable compared with if you do a superiority trial and non-inferiority trial, that's going to be huge. And also the other thing you will notice is in general, when we have a slightly, when we increase the alpha, the type 1 error, the sample size is going to decrease, okay? But you will notice here, in some sense, it's going to decrease slower when we increase 20 to 30%. And in some cases, it won't decrease, okay? So at the beginning, we also kind of feel this is weird, right? So this contradicts what we learned. When you have a higher type 1 error, you always have higher power. But in this case, it's not the case. The reason is because our power is not standard power. We have additional requirement. We require, for example, at least one selected dose is truly admissible, or all selected doses are truly admissible. So because that constraint, and there is no really so-called, very monotone relationship between the alpha and the beta. So in general, we notice that, when the alpha, the type 1 error is 20%, if you further increase that type 1 error, you won't have much power gain. So as a general recommendation for your trial, you can always start from a 20% type 1 error. Increase won't gain you too much. And because in that case, the power is a constraint and it won't decrease that sample size. Okay, so here is some practical consideration to this simple design. And in small samples, because the design itself only involves simple comparison, number of toxicity and efficacy with threshold toxicity and the efficacy threshold MT, ME. And in some cases, you will notice maybe higher dose have less toxicity or have less efficacy. So if the monotonic assumption, it's reasonable for the drug under investigation. And in this case, you will end up a kind of anti-intuitive scenario, right? So for example, the high dose, it's admissible in terms of safety, but lower dose, it's not. But in this case, because that's reasonable due to the randomness. So what we can do is we can first do isotonic transformation on the number of toxicity, number of efficacy, make sure it's monotone. And then we can compare it with the MT, ME threshold to make a decision which dose is admissible. And the second point is about the toxicity and add the futility and the safety monitoring. So this is the one point mentioned in the FDA guidance. And in this case, we can use intuitive Bayesian posterior rule to make the decision. So for example, we can stop the arm J for safety if the toxicity rate, given the data, the toxicity rate greater than a certain threshold. So basically in here, we can use the alternative value as the threshold. So if it is high chance greater than unacceptable toxicity rate, we're gonna stop that dose arm. And similarly in the second one, it's futility. If the efficacy rate, there is high chance efficacy rate less than our target efficacy rate, and then we're gonna stop the trial. And so this is a very simple, intuitive futility safety monitoring. And should we include in this monitoring are gonna depend on the availability of YT, YE, right? So during the interim, you wanna make sure you have enough data to make this decision. And also depends on the logistic. So for example, in your drug, you don't expect any acute toxicity. You are more concerned about tolerability after multiple cycles. And in this case, added safety monitoring may not that effective, especially given the sample size we showed in the previous slide. It's typically 20, 30 per arm. As long as your accrual is reasonable fast, And by the time at the interim, you probably no one has data available, so you cannot really have effective safety and futility monitoring. So that's why all those things have to take into account before we evaluate, should we add this futility and safety interim monitoring. And if you do, and I think in general, 1 or 2 interim are sufficient. Okay, so this is some numerical simulation results. And because our design is just guarantee, theoretical guarantee the type and error and the power. So the simulation is just to demonstrate that it should be achieved. Okay, so this is the type and error and power under the those 2 non and alternative toxicity and efficacy rate we showed in previous table. And so you notice here is we consider this 2 dose and this is 3 dose. And the first, the panel A is type and error rate. Again, the interesting thing and challenge, as I mentioned at the beginning, is because there's multiple non, right? Because we have multiple dose. So there's multiple non corresponding to the non hypothesis. So in here, we shows the actually type 1 error under different non. So as we said, because in here we control global non. So that's even in the worst case, the type 1 error going to controlled. And the different color shows the nominal type 1 error rate. The red is 10%, green is 20%, blue is 30%. And the red line, green line, and the blue line shows the nominal level. And you can see in all different cases, it's a type 1 error all controlled. Some are higher, for example, some lower, because it's going to depend on the non-hypothesis. And the bottom shows the type 1 shows the power, and you can see even in the worst case. So in this case, in this scenario, it's a higher power, this lower power. But again, it's guaranteeing all the scenarios under the alternative. It has a 80% pre-specified power. And you will notice here seems conservative, right? Because the type 1 error actually always lower than much lower than it's much lower than the nominal value. The reason is again because in here we also have a power constraint, and our definition it's different from a standard simple definition of power. So in this setting, to reach the power, it's the critical determining factor for the sample size. So that's why when we do that, in order to reach the power, global power, and the type 1 error actually becomes more conservative. So that's the reason. Okay, so. And this is just the shows for the under the definition of power 2. Again, the basic message is type 1 error, global type 1 error are well controlled, and the power it's guaranteed. Okay, so it's a simple design, but we still have to determine the sample size per arm, toxicity, and efficacy threshold. So that's why we developed software to facilitate the use of the design. So again, we put this at trydesign.org, and if you go there, and there is a section called dose optimization. And if you go there, so there's list different designs for the purpose of dose optimization. So for example, Bayesian 1-2, it's one of the efficacy integrated dose finding approach. So at the beginning, it's considered both toxicity at the same time, and based on the utility to do dose escalation, de-escalation. That's one strategy I briefly mentioned at the very beginning. And here is for the merit design. It's a 2-stage approach, right? Stage 1, you do a standard dose escalation. You can use Bayesian or CRM, do escalation, find the MTD, and then you select 2 or 3 dose into the stage 2 for multi-dose randomization. So that's where merit design can be used. And if you click it, and this is the user interface, and it's allow you specify how many doses you're going to randomize to, and what's the non and alternative toxicity rate and the efficacy rate. And then you give the software what's your target type 1 error rate, and what's your target power rate. So you're going to use power 1 or power 2. So again, the power 2 it's more relaxed. Typically leads to a slightly a smaller sample size, and the power 1 it's more stringent. And also you need to input other settings to optimize the design, because it's going to do the numerical search, identify the cutoff which going to control type and error and the power. And because toxicity data and efficacy data, a lot of time they may be correlated. So that's why in here you have to tell the software you expect correlation is positive or negative. What kind of expect correlation? But again, this is not critical, this part, because this part is going to use to generate, optimize the design. And if you are not sure about it, and you can use low correlation, even 0 correlation. And typically that's more conservative, okay, unless the truth is negative correlated, which may be not that common. And you tell software what's the number of simulation used for optimization. And if you do that, click calculate optimal design. So it's going to give you the design description. So essentially, again, so this design only we need 3 things: number of patients randomized to each arm, what's the toxicity boundary, what's the efficacy boundary. That's going to be used to determine should we reject none, which dose we should select it as the OBD admissible. And this software also allow us to include in toxicity and the futility monitoring. Again, as I said, this depends on the trial characteristic, some logistic consideration. And if you, in your case, if the endpoint and also allow you to do that effectively, and you can do that. And in here, it's you should input, when you're going to do the interim, right? So basically, this is a fraction of information in terms of total sample size. So basically, here it says I'm going to do efficacy monitoring in the middle of the trial when I finish half accrual. And the toxicity I will do more frequently at 1/3 and 2/3, I'm going to do a toxicity monitoring. And this is the rule we're going to use, Bayesian rule, as I just described. The only thing we have to input is the threshold, the posterior cutoff. And we did some simulation. The default value typically is a reasonable one, 95%. But it can be calibrated based on your trial. So it's a good starting point for you to calibrate. And so if you enter that and you run the software, in addition to a design description, it will also give you additional information regarding the stopping boundary for toxicity and efficacy. So for example, in this case, you will see because for toxicity, as I said, we're going to at 1/3 and 2/3 do 2 monitoring. That's why you notice here they have 2 boundaries, 6 and 10. And the efficacy, they only do monitoring at the middle. That's why I only show in the middle at 5. And you can, the second tab allow you to do simulation to evaluate the operating characteristic. And you can further calibrate those cutoffs if you want, and the type 1 error, type 2 error. And if you run that, for example, this is the output operating characteristic under the different scenario you entered. So basically here, it's where you enter the scenario, the truth. And this will report what's the type 1 error and what's the power. And it also shows graph of those regarding this type 1 error power. And I think, yeah, so this is the last slides. And this some additional discussion point. The one is for the purpose of dose optimization. In general, adaptive randomization, it's not particularly helpful here because sample size is pretty small. And before you have enough data to make a reliable adaptation, probably your trial already finished. And also adaptive randomization have some logistical challenge, right? Require real-time readout and a more complicated randomization system. And also introduce higher variation in potential bias. So if you really think one of those, some dose may be overly toxic or not effective, what you can do is you can do equal randomization, add 1 or 2 interim monitoring, and typically that's sufficient for small sample size trial, like in a multiple dose randomization trial. And also, this merit design can be used in phase 1 MTD funding design or OBD funding design, right? So as long as you feed in this design dose set, then that's okay. It can be used. And also it can be used to construct a phase 2/3 design. So you run the merit design, and at the end, you pick one dose, move forward. The only difference is probably in the merit, usually including the control arm. And in this case, you can continue that control arm and one of those arm into the phase 3 phase seamlessly. So that's the phase 2/3 design. And in here, we only consider binary endpoint, but continuous and the survival endpoint can also be developed under the same similar framework. Okay, so here is the reference, and I will share the slides with you guys, so you can always look it up if you are more interested in this. And thank you.
Thank you so much, Dr. Yuan. So actually, the slides are already up in the BSWG KOL website. We have a webmaster already posted slides there. So if anybody want to look up the slides, feel free to check our website. If you have any questions, please feel free to unmute yourself to state your question, or you can type in the chat. So far, I haven't seen any questions coming through.
Ying, it's Brad. Great job. I really liked your talk. I think, you know, the trialdesign.org site is a wonderful site, and I've used it many times myself. I'm a little, I've been reading this guidance document. In fact, it's proof positive. Here it is on my desk. So the guidance document you started out with today is exactly what I'm thinking about right now. And it doesn't use the word Project Optimus anywhere in the document, but it's all about Project Optimus, as you correctly identified. And you know, the parts that you quoted from the document are really, I think, kind of a sea change, right? I mean, to suggest that we should no longer be finding a single MTD, that we should find a range of possibly optimal doses and then test them in a randomized fashion. It's kind of blurring the line for me between phase 1 and phase 2. Is this a phase 1 design still, or is this what happened to all the concerns we used to have about carefully, cautiously increasing the design? Oh, is everything okay? Can we go on to the next design or the next dose? Or what if we're in 2 space? How do we increase the dose now? And there used to be all that caution about that. And now we seem to be diving right into a randomized design and powering. And are we still in phase 1?
Yeah, thank you, Brad. Yeah, thank you for your for kind of comments. I don't know. I think nowadays the phase 1 and the phase 2 the boundary are really blurred, right? So for example, even in phase 1, if they don't do randomized, a lot of phase 1 trial right now do a lot of backfilling patient, and in some sense that's kind of combined a phase 2 component into it. So I don't know. I think it's kind of more terminology, but for this part, I will call it a kind of phase 2, at least phase 2A, because you randomize and typically I think at least in the real trial I got involved in, at least we are randomized more than 10 patients, sometimes 15 30 patients. So I do think it's kind of in a typical range of phase 2 in terms of sample size. And also, traditionally when we have phase 1, phase 2, one important factor to distinguish that is what's the primary endpoint, right? So a lot of time when we say phase 1, we mainly do efficacy. In phase 2, we do some preliminary efficacy. That's our primary endpoint. But in this setting, as you point out, for optimizations, you have to consider some efficacy. Mm-hmm. It's almost kind of similar important as toxicity. So from that perspective, I kind of think.
So am I right in thinking that your advice to us then is that I should first use Boyne or something to kind of scope what the dose ranges are, and there I still want to use a cohort design. I'm not as worried about the randomization. I'm worried about scoping the dose space, and then I would use merit to do a preliminary efficacy. Is that the 2-step process you're endorsing?
Oh yeah, so I think that's the general approach. And actually, this slides, of course, this slides, it's kind of from some FDA presentations when they made about. Of course, they don't mention American design. Okay, so that's I want to make clear. They don't mention any design.
Sure.
So that's this two-stage sense. It's kind of I kind of align with agency thinking. Stage one is to mainly identify the safety set. And of course, you will collect some activity data, some efficacy data if it's available at that time. And then you.
I also have a similar line of question. This is Jay Lee from BMS, Ying. We communicate frequently about type Boeing 1, 2, and 2-stage design. And great to see you also develop merit and OBD randomization study. So as we just discussed, my first question is, this can be actually utilized in the phase 1 setting in the expansion course. And then the danger when we run this kind of randomization in the phase 1 setting, it's unblinded. And then there's a temptation, we look up the data continuously. So as we all know, we look up the futility multiple times. It definitely hurts the power. So my question is, I mean, unlike the phase 2 blinded setting, if you do the continuous kind of lookup, how much does that hurt the final power? That's a bit of concern. And I know also my second question is, people entering is probably beyond today's talk, but I mean, what's your thought? Well, how do we choose the 2 or 3 dose levels to randomize? And those are my 2 questions. Thank you.
Yeah, thank you. I can ask you your first question. So during the randomization, do we do a continuous monitoring or not? It's my made in my last discussion slides, and in general, I think 1 or 2 interim it's sufficient. The reason is, at such early phase, there's so much uncertainty, and the sample size is small, and it's very easy to make a mistake, right? So even we simulate the operating characteristic, and in that case, actually, we assume, strictly speaking, we assume it's binomial distribution. But a lot of time, actually, patient is much more homogeneous. We certainly have a dispersion sense over there in early phase. So that means we are quite a bit likely. If you set the cutoff or threshold very strict, it won't kick in because sample size is so small. Given each arm just 20, 30 patients. So that means virtually it's not effective at all. And but instead, it's the cutoff is kind of low, and then it's very easy to stop due to any big noise. So that's why my recommendation is, 1 or 2 interim. It's sufficient.
Yes, I absolutely agree, and then I hope that you can generate some kind of simulation and then guide, because I like to guide, for example, our clinical stakeholders. We don't want to look up more than twice, for example, and then I certainly provide them some quantified reason why not, and then that way we can guide them. We probably have to plan when we try to look up the data and then decide our decision. And that would be great to have that together with this tool.
Okay, yeah, that makes sense.
Right, I have to chime in that I feel all of us working in early oncology before had the same pain. Because it's unblinded study, we actually watch those patients one by one as they come in. Every new data points, we are generating new listing, new tables. It's a little challenging because there's actually more to do with the dose finding in a trial than I know what we are presenting here today is a more simplified trial design. They can change the pre-medication and switch the dose schedule, adding a little more safety monitoring. So it kind of mess up with the final results. If you understand what I mean, because there's so many things you can manipulate during this trial, the eventual results will be more confusing if there's too many hands in the manipulation part of the trial design. So I almost feel I really want to tell the clinical colleague to be a little more hands-off once the trial starts, especially this randomization part starts, so we have a cleaner data for our assessment. I do share the pain working in early oncology before.
Yeah,
Yes,
and also my understanding. Okay, this is my understanding when I read the guidance, and also went to those presentation by regulatory in terms of optimization. I think one goal they want to have is we have some data, sufficient data on some of those, so we can make a final call. So that's why I think you do a lot of frequent monitoring the stop arm too early. Based, for example, 6 patients you stopped, 10 patients you stopped. It's because I feel at the end, regulatory would like to, I just make it up, okay, 15 patient data, so they can make overall assessment if that's well justified, those selections well justified or not. That's why I don't think, you don't want it. Oh, of course, it's a safety issue. Of course, you should stop early, but otherwise, probably we should collect a little more data rather than stop too early.
Okay. Gary, I know you have a question, but Thomas actually typed in a question a little earlier. I want to see if Dr. Yuan can address that question. Thomas is asking whether you can elaborate when to use power 1 versus power 2. Do you have any guidance on that?
Yeah. So, for power 1, it's certainly more accurate, because it requires all the selected admissible set from the design, from randomized phase, are truly safe and efficacious, right? And that means you almost capture the best dose in your admissible set. But in power 2, we just require one of them, at least one. So it's likely there's some good dose you may overlook in that setting. The reason we do those is when we develop this design is we feel for industry. I heard a lot of people feel the main challenge is sample size. They want to get away as smaller as possible, at least from sponsor perspective. Although from statistician most of the time we don't agree. But there is a lot of decision-making people always trying to. Before we don't have to do this right now. Why we should additional 50 patients? Just slow down pipeline. So that's why when we worked on originally sort of power 1, it's statistically it's better, or even from a quality of trial, make better reliable decision. That's why we sort of can we further reduce that sample size. That's why we come with power 2. Again, I think in terms of how you choose, it should depends on your tolerance of false positive, right? And again, after all, in either approach, sample size is relatively small. It's just 20 to 30 patients per arm. That's kind of my recommendation. If possible, if you can afford it, to power 1. And if you are a small company, you really want to reduce sample size for power 2, it's also a reasonable approach.
So Professor, I have I guess a follow-up question. So I guess do you use the dose escalation phase to determine which doses are truly safe and efficacious? I mean, at what point do you or where do you get that information from?
So what we do here is, so here is power. Power is under the hypothesis, so we know the truth, right? That's how we control it. But in terms of, when we finish the randomized phase, should we leverage the phase 1, dose escalation part of data in those doses, randomized doses to make a more reliable decision. I think that's a separate question. But in my simulation and those things, I only consider randomized phase because it's a little bit tricky. Should we move that part of dose escalation part of data into here? And if you notice, is FDA guidance talk about the reason, the first justification for the randomization is they want relative comparable population through a randomization. I think if I do it, I will design this way, and at the same time, I will trying to do a kind of supportive analysis, including also the dose escalation part. Typically, you're going to have 6 or 9 patient, more patient, to see if the results are consistent.
Thank you.
Thanks, Gary.
Hi, Ying. Thanks again. Excuse me. Thanks again for the talk. I have actually 2 questions. So the first one is, with 2 or 3 dose levels that you're considering, it seems to me that it's a bit easier than if the number of dose levels that you're considering is much larger. And so in the sense that, at least if you have a large number of dose levels, there are more errors that you can make. What's the largest number that you've simulated in terms of numbers of dose levels? And then and at what point does it sort of start to break down, where you just really need a lot more patients in order to find this range of optimal doses? And then the second question is just, I assume you're using uniform priors when you're doing your your posterior calculations. But you do have information from the first stage where you're doing this dose escalation to find your capital J, your number of dose levels that you want to look at. So is there a way to incorporate information that's collected in that first stage to have a more informative prior in the second part?
Yes, thank you, Gary. Yeah, it's yes, I'm in Johns Hopkins. It's kind of a misleading. Anyway, so for your first question, we evaluated. I think in the paper we didn't report dose level 4 because I think we tried it, but I don't remember exact results, because my understanding is in practice it's very, very rare more than 3 doses, because again, at least my interaction with the industry, and is that right now they already complain this extra step require additional sample size. So I don't think it's they will do more than 3 doses. But I agree, it's interesting from a statistical design perspective. Yeah, we can go back, take a look, see how that things change. That's for your first question. And for the second question regarding how can we factor the the dose escalation part data or information into this multi-dose randomization part decision making. For the threshold and toxicity and efficacy threshold for decision making, for those part we don't make any we don't use any prior distribution. It's just strictly calculate those probability under the known and alternative hypothesis, as we showed here. So this is just binomial, and we can exactly calculate what's the probability. But for toxicity and efficacy monitoring, yeah, it's true. We use posterior probability make a decision. So for that one, we didn't take that in account. But I think it's a good idea, at least for toxicity, we should use escalation part of the data to calculate that probability to make more reliable interim decision.
Thank you.
I see two questions online from Fujin. One, the second question is similar to Gary's second question. I'll read the first question from him. It says, you use tox range of 0.2 to 0.4. That's kind of wide. That may cause concern in some clinical colleagues. So he's asking, have you ever tried the tox range 0.25 to 0.35? Any meaningful impact on the outcome?
Yeah, so yeah, that's a great question. So one thing I want to clarify is it's not say 0.2, 0.4 acceptable. So in here we set non-alternatively. Alternative is to control the property, type and error or power under those 2 point value. And then you can imagine if we put it there too close, basically when you control type and error and the power at the same time, it's unlikely to happen. So in here, the 0.2 means it's very safe. And in this case, I don't want to make any high type and error. And the 0.4 means it's very toxic. In this case, I don't want to select that dose. I want to guarantee the selection of that dose is very low. So that's the interpretation of those two values, which should be, quite a distance from each other. So remember those two values, it's just used to quantify control type and error, right? So if you want to control 10% or 20%, certainly you want to put it in a relative extreme case. So you at that point you control low. Of course, if it's true value, it's between the type and error or power will be between, and that's the reason and interpretation of those two values.
Thank you. If anybody else have any questions, feel free to unmute yourself.
Ying, can you also comment with your experience how you to recommend to choose the 2 or 3 dose level to enter this kind of randomization trial? I mean, based on many of your tools like a Boeing 1, 2 or Boeing. I wonder what's your experience and what's your recommendation?
Yeah, so the first day is in general. Okay, here is my thinking. So in general, it's feasible doing such randomized multi-arm kind of if we call phase 2 trial, definitely give a statistician more confidence because it's randomized. They will have more data and we can make better decision, select optimal dose. So if possible, that's also the FDA guidance said, if possible, you should randomize. That's language they use, and if possible, do that. And I think if you do that, the only thing matters is at the beginning how you identify the multiple dose to move to this second randomized phase. And I think for that dose escalation and set into this randomization phase. I think that Boeing or Boeing 1, 2 both can use. And I think the difference is this: for the Boeing 1, 2, it's also require a specify some efficacy or at least activity endpoint to make it that, based on desirability, make dose escalation, de-escalation. So it's a little bit more involved, and also you need a relative quick efficacy readout before you make that decision. But it's compared with the DLT-based escalation, it's more efficient because at the very beginning, it's already targeted to dose, which most likely to be the OBD. So you can imagine, actually dose escalation, I always think dose escalation is a process of patient allocation. Not really, of course, at the end, you will pinpoint that. But underlying the mechanism, especially from treating patient perspective, it's kind of a patient allocation process. And then you want the end product is the design can allocate a patient around your target dose. So that's the thing. And I think for Boeing 1, 2, because at the beginning it's take desirability, that's why it's going to concentrate most patient around the OBD. And that will inform you better to select the 2 or 3 dose into the randomization phase. And for DLT based, it's ignore that information. But the advantage is it's much easier to do, and also the DLT it's quickly observed, and you can make that it's simpler. So basically,
Thank
you. I'm waiting to see if there are more questions coming in.
So Dr. Yuan, you just this is Fujian, just a follow up to my question as as before. About the 0.2 to 0.4, is the width matter? Does the width matter to the outcome, or the location matters more?
you mean 0.2, 0.4? Okay.
Yeah, like two different.
Width.
Yeah. So so that's toxicity, right? You talk 0.2. Those value gonna determine again, the reason we have those 2-point value is to control type I error and the power for that purpose. And if you make it small, so the consequence is the sample size going to be pretty big.
Because I'm sure when other people, non-statisticians, see those numbers, they can be very concerned.
You mean concern in terms of sample size, or
no, the high limit, the treat that 0.4 as a high limit of toxicity we can tolerate.
Oh no, it's not tolerate. Okay,
I know,
so
I know.
0.4, yeah. 0.4 actually, it's a very toxic case. In this case, we want, for example, control the error rate less than 10%, or that's the interpretation of 40%.
I know, but I'm trying to be, the salesman. I want to
okay.
sell this method, then I need to answer that question.
Okay.
Many people could be concerned, including FDA, reviewers. Sometimes they just see that one as too toxic, you cannot use.
Yeah, we don't use it, right? So it's just the basically you can tell that you have two hypotheses, right? The non-hypothesis 20%, that means acceptable, and 40 is unacceptable. I think it should be the reason actually we focus on that simulation is because by convention people feel 30% is the upper limit. That's why we one it's a lower, we want the lower one, it's always almost always acceptable, and the higher one it's rarely acceptable. That's the rationale we use 0.4, 0.2.
Yeah.
And Dr. Yuan, I see Brad has a comment asking about the reference paper. I don't know the last slide in your slides deck had the 2 reference paper. Is that for merit or point?
Yeah, one for merit, the other is for phase 2/3. Yeah, I can add. The reference and send to you, Fred. And probably you can update the slides.
Sure, we will do that. Yeah, I remember there's 2 reference papers at the last slide.
Thanks, Ying.
Thank you.
Any more questions? If not, I do really want to thank Dr. Yuan for this fantastic talk, and thank you all of you guys joining us today for this KOL seminar. I hope everybody have a great weekend.
Yeah, thank you.
Thank you.
Thanks, Frida.
Thanks. Bye.
Thank you. Bye-bye.
Bye-bye.
Bye.